Hi, my name is Blake Sweeney. I'm the project leader for Rfam and RNA Central. And today we're going to walk through what's in Rfam, how you guys can get access to it, and some examples of some families, and then a quick demo of annotating a viral genome, SARS-CoV-2, with Rfam families. Please, as we go along, put any questions you have in the chat. Some will be answered in the chat, and then some will be answered at the end in a Q&A session. So thank you. And so who's with us today? So Emma Cook, she's a software developer for Rfam. Nancy, she's the curator for Rfam, so she's responsible for all of the families. And myself, I'm the project leader, and then Sam Griffith-Jones, who's actually the founder of Rfam about 20 years ago. Because Rfam, and so what we're gonna go over just the outline for today. A quick overview of Rfam. So what's in it, what's not in it. Look at the website, how you get the data, some examples of families, and then the demo I mentioned. So yeah, start with the overview. Rfam's 20 years old now, and the first paper was published in 2003, actually, in Jan 1. So it's got a long history. We have this little page you can go and look at and kind of see what the point of Rfam was, why it was created, and all these things. It's kind of neat to hear from all the people, from many of the people involved for all these years. So it can be, I enjoy talking to them at least. So. Right, so now let's look at Rfam. So the current release is 14.9. It's got just over 4,000 different families. Rfam is a database of these RNA families. So it's not of individual sequences, but it is of a family. And so what is in a family? I like to think of it as being made up of 4 different things. So an Rfam family includes a multiple sequence alignment of the RNAs that someone believed to make this structured non-coding RNA. This alignment comes with a consensus secondary structure that represents the base pairs in the alignment, which is then used by our curator, Nancy, to build a covariance model, which is how you find these RNAs in a genome. And it also includes a lot of annotation and curation work. So our families have Wikipedia articles. Some of them are generic, so say all the microRNAs, or some of them are very specific, so a particular microRNA. Someone may have written an article about that. We also have Go terms, so gene ontology terms and sequence ontology terms, to give an idea about the function of this RNA. So it's not just the alignment in the models. There's a lot of curation work that goes in as well to give functional information. And so what do people use Rfam for? So genomic annotations, as you may guess, because that's what we're talking about today. So for example, Rfam Ensemble uses Rfam to annotate all small non-coding RNAs in genomes. It's also used as a reference set for non-coding RNA families. So sometimes people want to know what non-coding RNAs exist. They'll look at Rfam. Or what is the secondary structure of a particular non-coding RNA? They will look at Rfam to see what we say it is and use that. And this leads to kind of one of our third major use cases, which is as a training set for algorithm development. So sometimes it's I'm sure you guys have heard about AlphaFold and all these things, but using machine learning to predict various 3D or 2D information about a molecule. And for RNA, what's often used as sort of the source of truth is Rfam families. So the 2D we propose and those sort of things is often used as the source of truth. So one thing that is very important to know about Rfam is we're heavily powered by this software called Infernal. So when I said we build a covariance model, and then that's used to annotate genomes, Infernal is what does that. There's a set of programs that does it. It can also be used to build an alignment of hits of a particular family to a genome. And we're going to show you a brief how to use Infernal later today. And so I kind of talked about we have 4,000 families, and you might wonder, how'd you get them? Where'd they come from? Alignments are usually taken from the literature, or sometimes a user will reach out with an alignment and say, I've published this non-coding RNA, please add it to Rfam. And so that's basically where we've gotten a lot of them. We also have grant projects, which will focus on creating some specific families. So you'll hear about miRBase later, as well as some families that are created working with the European Viral Bioinformatics Center. So kind of important thing to know is what's in Rfam. So about 35–40% of the families, so just over 1,500 of our 4,000, are microRNA precursors from miRBase and Sam Griffith-Jones. The next largest group are SNO RNAs, and then sRNAs, which are basically bacterial regulatory RNAs, is what Rfam calls sRNAs. That's a nice graph. You guys can look at it if you want to see all the types. But then what does Rfam not have? We do not have whole long non-coding RNAs. Some regions in them may be candidates for inclusion into Rfam, but not whole long non-coding RNAs. We generally don't include small RNAs, so less than 50 is kind of the cutoff. An RNA that is only present in a single species is also not usually a candidate for inclusion into Rfam. And then finally, there's this sort of last group of things we can't build a good model for, which can be all sorts of reasons, some of them kind of boring technical reasons, and maybe some of them that have a different source. And so I suspect many of you might be slightly disappointed to hear that we don't have whole long encoding RNAs. Unfortunately, EBI has another resource, RNA Central, which is about collecting all RNA sequences, which will include long encoding RNAs. And some people do use this as, a source of truth to help annotate long encoding RNAs. There'll be a webinar about RNA Central in September. And if you're interested in that, or you want to hear about that, feel free to sign up and maybe put a question in to let us know that you would, if you have a particular thing you want to hear about on long encoding RNAs, let us know. We'll see what we can do. So with that, I will switch over to Emma, who will give you guys a tour of what's in Rfam and how to get it.
Thanks. So I am going to take us on a whistle-stop tour of the Rfam website. So once you navigate as far as rfam.org, you'll be greeted with our homepage. You can see the main message on our homepage there is our most recent release, the date, number of families. At the top of the page are quick links to the most useful parts of the website. Again, at the bottom, you can see there our Getting Started section of useful links as well to get you going with the Rfam website. And right in the center, you can search and browse Rfam straight away from the homepage. So I'm going to take you through a search of Rfam and kind of what you can expect to see from a family page. For example, if we search for 5S or RNA, this is a straightforward text search. You can see a list of results there with families that include these terms in their name. On the left-hand side, we can see that we can filter by things like entry and RNA types, and further filters available to us there. But I'm going to go ahead and imagine that we've clicked on that top result there. So for a 5S ribosomal or RNA, you're brought to a family summary page. This is linked to our Wikipedia pages. It gives a good overview of the family and kind of information that you would need to know, want to find out. And on the left-hand side of the family page, we have a series of tabs with further information and some more in-depth information related to this family. We're going to take a look at some of these tabs. The first one there is the sequences. So you can download all of the sequences available to a family, or you could download individual FASTA files. Sometimes there may be too many regions to download for a family. It might be too large. In that case, you can simply reach out to us, contact us, and we can usually get those files to you without much issue. Another thing we can take a look at is the secondary structure for the family. You can view the secondary structure in a variety of representations. What we can see here is the current Rfam structure compared to the RScape optimized structure. In this example, we can see that the highlighted green areas are covariing, statistically significant covariing base pairs, but there's a number of different secondary structure representations available to us there. Something else we can check out for the family are PDB structures. So for sequences that have a structure in the protein data bank, we map that information between PDB and Rfam. So we have a list here of all PDB IDs linked to this family, and we have links out to the website so that we can find out more information on those structures. For example, if you were to follow the PDB-E link, you can find more structure analysis information. You can see things like this visualization tab here, where we get more information on the chain or the residues that are mapped to Rfam domains. Another thing we can take a look at is the species distribution for a family. So here we have a modified sunburst for this particular family. We can also view the distribution of this family across species in a tree format. There's a number of different options available on that tab. And another interesting thing we can take a look at is the publications tab. So here we can see open access publications that mention this particular family in their title, main text, or abstract. This is all powered by a text mining pipeline called LitScan that is run by the RNA Central website. And it has a lot of different features that we can use to filter through publications and find ones that are most relevant to what we're interested in. Another very useful and very important part of our site would be our FTP section. So again, if you're going from the homepage, the FTP side is available as a link from the top of the page. And that brings you through to all the files that we have available to download. So as an example here, if you navigate to our current folder, you can find all the files related to our current 14.9 release. These are things like database, FASTA, CM, seed files. We also have our older files for our 14 releases and for older major releases going back a little bit further there. We also have details on the website of how to connect to our public database. So all the connection details are available to you there. Also, so once you connect through to the database, you'll be able to access a whole lot more information. An example here is of the family table, and we have some example queries available on the website of things you might want to run or try out to find more relevant information to you to extract some further info from the database tables. Another very important section of the website is, of course, our help section. Again, if you're coming from the homepage, the link to the help section is right on the top there, and you'll be brought through to view things like FAQs, glossary, again, some of those SQL queries that I mentioned, and also very importantly, how to contact us. If you have any further queries, suggestions, comments, all of that information is available on the Rfam help pages. Now I'll hand over to Nancy.
I'm going to go through some examples of the kind of microRNAs that we are working right now to improve in the database, but also this is a good reference of what kind of non-coding RNAs Rfam is representing. So the first example that I want to show you is related to microRNAs. As Blake already told you, we have around a 40%, a little bit less, of the size of the database that are representing microRNAs. And this is something that is would have been increased in recent releases, in this 40 point whatever releases. So because we are now working with the data that is in miRBase. So by the time that we compare both databases, miRBase, that is the microRNA database, versus what microRNAs were in Rfam, we have around a 40% of the amount of microRNAs that a miRBase was representing. So by now we have like 1,500 more microRNAs in Rfam, but an important thing is that we are also reviewing the microRNAs that we have in the family. So this is an example, is miR17, and what you're seeing on the left is the previous Rfam alignment, and then in the center you're seeing the new alignment. So this has a different secondary structure. And when we work with the data set of genomes, searching for this microRNA, we can figure out that the covariance is really good represented for the secondary structure. So this is a kind of improvement that you can see in the families that are representing microRNAs. Then, as also Blake said, some of these Rfam families are taken as the true for the secondary structure of a non-coding RNA. But the truth is that most of that models come from secondary structure prediction. So this make a situation where we actually have to review with 3D structures. So most of the Rfam families were created before we have information about the structure. But now we have found that more than 130 families in Rfam are also having information in PDBE, the protein data bank, about the structure. So we are reviewing each of these non-coding RNAs and making any update that has to be to correct the secondary structure. So I'm going to go through two examples. One of these is the family 162, that is the SAM ribosus. And you can see that in the previous secondary structure for this family, we have a big loop in the center of the secondary structure. After we review it with the structure that was reported in PDBE, we found that there were base pairs that were not reported. And we also have now information about regions that are not well reported for a secondary structure prediction. There is this thing that we call here PCA, that is a pseudonode. So this kind of information can be integrated now and make a more accurate structure for the SAM ribosus. So I'm going to show you another ribosus that has this already review and update secondary structure. So you can see also in this one how the center of the structure was not well represented. And that can happen because the secondary structure predictions cannot deal with this kind of base pairs that are from very not near regions. So sometimes it's not easy to be predicting that these base pairs are there. But now we can actually add and correct these base pairs with the 3D structure information. Then we also talk about the viruses, and we have been updating a lot of the information that we have with viruses and creating new non-coding RNA families, because there are a lot of viruses with RNA genomes, and in the end, they have secondary structures that are conserved. So this is a work that we are doing in collaboration with the European Virus Bioinformatics Center. And some of these examples are these ones, flaviviridite RNA families that are representing the 3' UTR of these genomes in flaviviridite families. So with this, I'm gonna go and give back to Blake to the rest of the seminar.
Thank you, Nancy. So now I'm going to annotate a viral genome, SARS-CoV-2, using Rfam to find all the non-coding RNAs in it, and I'm going to try to do it live. So see how it goes. But just to say, what do we mean by finding what do we mean by annotating a genome? So genome's just a bunch of letters, just a big long sequence. Can't really tell what's in it. you run Rfam, it'll tell you where there's a hit, and then you can look at Rfam to see what that family is. And so today we're going to use Docker and Infernal. If you're not familiar with Docker, it's a really excellent tool. I'm not really going to talk much about it, but there's a ton of tutorials out there about Docker. I have a couple links at the end of things I looked at that seemed helpful. And so if you're new, I really encourage you to spend a little time to learn about Docker. Okay, so maybe if someone can drop that link in chat, it would be great. If not, it's in the slides. And this links to our tutorial that we've developed to help do this. That page also has a link to a paper we've written about using Infernal to annotate genomes, which is more comprehensive. And actually, that paper contains all sorts of things beyond just that. So we encourage you to take a look at it as well. So, all right, so here's what here's a terminal shell. And what we need to do is we need to start out by getting the latest tutorial image from Docker. I already did that, so basically this didn't do anything, but it's just there to show. And then we start up a shell inside Docker to test things out. So let's take a look at what we have. So we have rfam.cm, this file here. This is all the covariance models that Rfam provides. You can download, you can get that from this image if you would like, but you can also pull it from our FTP site that Emma showed you. There's an rfam.cm there, which is what this includes. And then there's also this rfam.clanin. So many Rfam families are very similar to each other. Maybe it doesn't surprise you that all this, say, small subunits look kind of the same because they all have fundamentally a similar structure. And so clans are a way that we sort of deal with that duplication of matches of hits in the genome. And so we also encourage you to get that clanin family. And then finally, virus.fasta is the genome for the SARS-CoV-2 virus. If you want to take a look at it, it's kind of a regular FASTA file. And so the command to run is, I have to grab it again, sorry, is this long command. Or first we start with cmpress, rfam.cm. So before you annotate a genome, you have to, before you use the rfam.cm, you have to run rfam or cmpress. You only need to do that once. So let's say I had 100 genomes to run. I do cmpress rfam.cm, and then we wait while this finishes, and then you can annotate each one of those genomes. You don't need to do it each time. And one thing that maybe some of you may or may not know, since I'm running this on Docker and I have a newish Apple computer, it's way slower than it should be. In fact, this is, to give you an idea, rfam press would normally take 30 seconds or less, and this takes closer to a minute. This is a downside of running Docker on your newish Apple computers. It's not the case for you. You don't have to worry. And then this command, which is maybe scary, but I'm going to explain it as it runs, will produce annotations of all non-coding RNAs in the SARS-CoV-2 genome. So I'm going to try to do a little basic Unix explanation for everyone while this runs. So we're running the command cmscan. This is part of the Infernal set of tools. So a lot of Infernal tools start with the letter CM. So cmpress, for example, cmscan is another one. And then there's these things that start with dashes. There's a whole lot of those. Those are what are known as options in Unixy language. And then there's these two things at the end called arguments or positional arguments, depending on what you're reading. And so the rfam CM is the set of rfam families, all the CM models, covariance models, and the virus.fast is a sequence we're going to analyze. So cmscan is scanning the covariance models against that sequence to find hits. Now, these options, some of them are important. I'll kind of walk you through what they are. So the first three, to start with, we recommend that you always use them. So -- cut _ GA means use the thresholds that the Rfam curator found to annotate the genome. And so as the family is built, a curator looks at how the model does and says, well, below this are hits that I don't think are real because it's the wrong phylogeny, because the hit doesn't look like the original alignment, whatever reason. And so you want to use cut GA to make sure you always use that. --Rfam speeds it up and runs it in the same way that Rfam does to annotate genomes. No HMM only means that basically it doesn't run an alternative model and really will actually definitely always use the thresholds correctly. Finally, this clan in option and oskip option are also recommended for dealing with duplications in the families. So we talked about, say, if there was a small subunit in the SARS-CoV-2 genome, there isn't, but if there is, the bacterial one, the eukaryotic one, and the archaeal one all tend to match each other or kind of overlap. And this will prevent those overlaps from being shown because really you wouldn't expect all three, you can only expect one. And then finally, these last options, format2 and -output.txt and table.txt control what kind of output is written and where. And so I will show you what is there now. So if we look at output.txt first, this is all the hits, not just the deduplicated ones, all the hits that are in the SARS-CoV-2 genome. So there are 7, including duplicates. It includes information about where the hit is, how well it's scored, and the alignment of the model to the genome. And so you can look at all of them. Another very useful file is the table out file, which contains only the deduplicated ones. So in this case, there are 3, because as it turns out, some of those hits are families within the same clan, and so it only takes the best match in a clan. And so this gives you the information of where in the SARS-CoV-2 each match is. Another thing that is often needed when annotating genomes are bed files or GFF files. And so this, you can produce either one from a table out file. And so, so this tutorial includes a little script. I added table out to bed, which will take these table out results and turn them into a bed file. And this is available to you. It's a very simple bed file. It can be extended on. We can produce GFF files if you want something richer. I'm actually very happy to write that code. It's something I'm probably going to do anyway because it's so useful. But you can use the script yourself, and then now you have a simple bed file. You could take it to any of your genome browsers online. They basically all use bed file or accept bed files, or a whole host of other tools use bed as well. And so that is the tutorial. So just to say what we've covered, we did an overview of Rfam. We showed you guys what data is there and how to get it, and looked at a few families, and then annotated a SARS-CoV-2 genome using the Rfam families, produced a bed file as well. And so here are some useful links. This, links to Rfam, RNA Central, our help pages that Emma mentioned, the tutorial that I just ran everyone through. This paper, non-coding RNA analysis using the Rfam database, which has all this stuff and more. The Infernal help docs, if you're going to use Infernal extensively, I really encourage you to take a look at them. It's pretty well documented. Some links I thought that were nice for Docker, but there's really a huge amount of information out there. Just Docker will give you, tons of reading. And now it's time to answer questions. So if you have any questions, please put them into the Q&A and we will answer them. I will read them out and pick on whomever seems to be the best person to answer them. So there's a question, is it easy to know which species are available? So do you mean which non-coding RNAs are in which species? Or so we do have pages in Rfam that for a particular genome that says, say, the human genome, and it will provide you a link to go to all the hits that are in that particular human genome in Rfam. That is available on our site. as well. You could also mean, is there an easy way to get the non-coding RNAs that come from a species? And that's a different question. So what species are in the repository? So we use the UniProt reference proteome set, and we get the genomes from that. I believe the last time it was updated was about 8,000 species. So they have a paper describing it, but very briefly, it's meant to be both representative of all the sort of the diversity, but also deduplicating a bit. So it doesn't have, for example, all the E. coli genomes, because there are an incredible number of them, but it does try to have the important ones that people study. What machine learning? So we don't use machine learning to predict the 2D structures. The 2D structures are generally given to an author. They may be predicted in the authors, but I don't think we have a large set that are predicted by machine learning. There are 4,000 families, maybe one is, but I don't think that's a significant portion of Rfam. Can we annotate in amoeba? Yes, if we had an amoeba sequence and we ran that CM scan, we would have the annotations in amoeba. It's the same command regardless of species, and you will get matches. So yes, it will. I don't know what non-coding RNAs are in amoeba. I'm not familiar with amoeba in general. Can it take bigger genomes? Yes, it can scale to larger genomes. They take much longer to analyze naturally, but that is possible. We used to annotate plant genomes, which are fairly large, as far as I'm aware, and it works. Obviously, it's slower. It requires more memory. Sometimes there are some things you can do to cut that down if it's a particular problem. If it doesn't work in your genome in question, maybe reach out to us. There's some very specialist options in Infernal that can do things to make it more efficient. George is asking about the online. So we do have a tool where you can upload a sequence and we will analyze it with Rfam. Yeah, that does have a limit. We have a finite amount of resources, and so we do limit the size of sequences that we will run on our servers to annotate as well. How do I identify false positives, which I think is similar to contamination? Okay, good to know it wasn't just me. Okay. So false positives are usually, but not always, lower scoring hits. So that's one thing. You can look at them and see if the alignment looks right. If there's a disruption in a site that you think is essential, so for example, the seed sequence of a microRNA and the seed sequence doesn't look right, maybe that's a false positive. We do try to make the models as accurate as we can to prevent false positives, but it is kind of inevitable that there will be some. The visualizing contamination, Sam, did you have an answer for that, or should I just say something?
I can do. So actually, as a sort of an anecdote on that. So one way in which Rfam hits could be really useful, actually, for seeing contamination within it's within sequences that are sequenced, genome sequences that are sequenced. So actually, in the early days of Rfam, there were surprising hits to a bunch of models of bacterial RNAs in the draft human genome sequence. And I suspect there probably still are, because there are various bacterial vectors that are used as sequencing vectors. So as part of the sequencing process, there are sequences cloned into bacterial vectors, and sometimes those little bits of bacterial vectors end up in, and even bacterial contaminated genomes end up in bigger genome sequences. And so actually, you can sometimes find hits to models of the wrong species or even the wrong sort of kingdom in genome sequences, and it's an illustration of contamination. So that's kind of a neat anecdote that shows how hits to RFAM models might be really useful for spotting contaminated samples. There was actually, I wanted to say a little bit more about a question that Nancy answered in the text as well. So somebody asked, I'm trying to find it again. Somebody asked, how does Infernal represent both sequence and structure conservation at the same time? And so that's actually a really quite a kind of technical question. But as Nancy said, what Infernal is doing is building covariance models. They're also called stochastic context-free grammars, but we tend to say covariance models because it's much easier to say than stochastic context-free grammars. But they're a statistical model that represents, as the question asks, both sequence conservation and structure conservation within the alignment. And so you have an alignment that you can train a model on, and that multiple sequence alignment is of sequences that we know are related, non-coding RNA sequences that we know are in the same family. And so then you can see conservation at the sequence level between the sequences. But that alignment is also annotated with a secondary structure, so a base paired secondary structure. And I'm not going to go into the technical detail because I'm going to quickly get lost if I do. But the stochastic context-free grammar is able to represent both the conservation at the sequence level, but also how that conservation maps onto how the RNA folds up, and so where the base pairs are formed in the RNA. And it turns out, actually, for a reasonable alignment, a reasonably deep alignment, that Infernal gets about the same amount of information out of sequence conservation and structure conservation. So it turns out that sequence conservation and structure conservation are roughly equally used in terms of getting a good score for a hit to a model. As I say, if I were to get into the detail of that much more than that right now, then I'm going to quickly tie myself in knots. But there's a bunch of papers from, first of all, from Sean Eddy, who wrote the first version of Infernal, which describe the application of these models to RNA, to finding homologs of structured RNAs that do go into it in more detail. And while we're still talking, I'll try and find some of those papers and stick them in the chat.
Thank you, Sam. I can say that those contaminations you see in eukaryotic genomes of bacterial things did not go away. As of at least a couple of years ago, there were bacterial riboswitches in the C. elegans genome, which I'm suspecting were contamination. Seems unlikely. Yeah, they still exist. And also to just expand that slightly, we do actually track or suggest what domains of life each family is found in, using kind of what's in the seed and the majority of the matches to the model. And that's also available, so you can use that to see sort of any really gross, large scale contamination, significant contamination like that. And then can you use fungal genome sequences with gaps to annotate? Yes, fungal sequences shouldn't be a problem, and can certainly use them like that. The gaps might be, but there's some things you could imagine doing with the gaps to work around that, either just cutting the sequence at each gap, or maybe something else smarter. How can we represent the output from the files in a manuscript? So I love secondary structures. I think they're great to look at, and so that's one of the outputs is a match to a model and the 2D. So that's one way. If you're talking about, say, the statistics of how many things there are available, that's, we had this many matches, and we had across these things, and so you can show them that way. Yeah, Nancy, do you want to answer that more?
Yeah, I've been thinking about this, about secondary structure. We use something that is called RScape to create secondary structures from alignments to have the visualization. So if you have an output with particular sequences that are an alignment, then you can use RScape actually to create a secondary structure model as the ones that we have in Rfam, and then publish that. I suppose that's one of the best options.
Yeah, would depend specifically on what you want to represent. I mean, maybe Sam might know a bit more about that. I think he's done some of it, but yeah, I think the answer might be like, do you want the overall statistics, or do you want, you know, a particular match, like exactly what you're trying to get? We might change that. Unless Sam has anything out there.
So I'm not sure I entirely understand, but I think some of your things about secondary structures, mapping information onto secondary structures is a really useful way of showing these things, I think.
And so there's a question. So basically, it seems like you're expecting a match to some particular RNA, and you're not getting that match. And so there's potentially something, you know, either the model isn't tuned right, or it doesn't have the representation of variation that you expect, and so you want to find homologs in that case. Okay, so I'm going to ask Sam again this. I have an idea, but I think Sam's done this, so he would probably be a better answer than I do.
Well, so one thing you can straightforwardly do, when Blake when he showed the command line, he explained the Unix command line that he was using to run the Rfam models against the genome. He used an option, cut GA, which is so what that does is it uses thresholds for each model that have been curated by the Rfam team. So by the guys here, by Nancy predominantly, I would guess, but probably also Blake and Emma. And so that's a threshold that the Rfam team have determined. Okay, hits above this score are real, and hits below this score are likely not to be real. But you can actually run that search command with your own threshold. So you can I think you can set an e-value threshold there, Blake, is that right still?
Yep.
So you can choose to run with a lower threshold essentially to try and see if there are any hits which might be, you know, relatively low scoring below the threshold that the Rfam team have curated for that model. But perhaps in your case, if you're looking at a particularly weird and wonderful organism, perhaps you know, you're finding you want to find some low scoring hits because you might have a particularly divergent match. So that would be the first thing that I would try, and a relatively straightforward thing to try. Much more complex and difficult would be to try and get into improving the model yourself, which is not impossible, but it is a different kind of task. And if you were interested in doing that, then I would suggest you probably talk to these guys about that.
This brings to me an idea also. Rfam has plans, and this is kind of models that are representing the same family but in different phylogenetic groups. So that's why they are in a clan. So if you are working with a genome that is very far from the ones that maybe are represented by the reference sequences, the seed alignment, then you actually need to kind of go to get homologous to that particular far genome from the ones that we are representing, and creating again an alignment that makes the covariance model the model that is representing actually that family. And if you do that, please let us know so we can include it.
There's a question, have there been cross-validation of secondary structures to experimental evaluations? Do we receive a reference page for the origin of the secondary structure? So I can answer that a little, and then maybe I'll ask Nancy to expand if I missed anything. So the secondary structures are generally provided to us by an author, and so they hopefully have some experimental support or some support about them. In addition, and we do track where the source of a 2D of what the secondary structure is in the Rfam families. And that is available actually on our webpage in the curation tab if you're interested, and can be fetched by an API or downloaded in various ways. But maybe Nancy can add a little bit more, like you know, about the 3D and other practical. Yeah.
The truth is that the most of the non-coding RNA representations, secondary structures that we have, come from prediction, secondary structure prediction. It doesn't mean that we make the predictions. It's more like someone that actually find that this particular non-coding RNA has a function also go and make the secondary structure prediction and all the other work. For some of these non-coding RNAs, now we have this 3D information where we can confirm, update, correct with the real 3D structure. Also, one of the tests that we make to evaluate the secondary structure are review the covariance between base pairs. And this is something that we recently do for all the families with actually RScape. So yeah, we try to keep update if someone have biochemical test about the secondary structure and are reporting the secondary structure because they have an inline biochemical experimental that reports actually which is a real base pair and which is not a real base pair. It's going to be there. But yeah, by now we are kind of just sure about what 3D structures per se is giving for us non-coding RNA.
Yeah, I will say it'd be great if everyone tested the experiment, the experiments we showed what the 2D of every Rfam family was. That'd be great. It's a pretty large project, and I don't think that's likely to happen. Well, maybe we'll get a grant for that. That'd be cool, but it's a big project. Okay, and so the question is, suppose you have a clan of related species, but with structurally similar, but sequentially dissimilar non-coding RNA sequences. Can the covariance model capture the alignment or the structure, even though the sequence in the database is not the same as the one in the genome? For example, between, I'm sorry, I can never pronounce species name, Fliperserum mitochondrial rRNA and Gandhi mitochondrial rRNA. So Infernal should be able to build an alignment of them. If they're, I don't know how highly diverged the species are, so I can't tell you how good they would be. Yeah. Nancy?
No, I think the idea is if they are sharing the structural similarities, even if the sequence is not that similar, we are expecting that they are part of the same family.
Yeah. So you should be able to run the program CM align with those particular sequences, and it will produce an alignment. And then you would have to evaluate if it's, good enough or suitable for your purposes. But yeah, so for example, all of the bacterial sequences are in a single alignment, and that's, while structurally similar, are highly sequentially diverged.
I think I can answer the next one also online. So can you better explain how does Rfam carry out the evaluating and refining of results after it's being searched for? So we have first something that we call the reverse sequence set of a reverse sequence that appear in the results. And this comes from a set of genomes that are customized, so are not real genomes. And if we have a hit in that genomes, that means that we start to have false positives. So that's the first cut that we get. Then we review where do we have the change in the phylogenetic representation for this family. And whenever we start to get things that are not represented in the start alignment, the seed alignment, we also make a cut. And a third level of reviewing is actually to know where all the sequences that come from the fair alignment are. So if we are missing sequences from the seed or missing or getting too much sequences after the seed, we can go and make a more detailed review about which is representing that. So that's more or less the way that we go through carry out the evaluating and refining of the results. I'm not sure if that answered the question.
Well, so can I add something to that, Nancy? So what Nancy's just described there is the process of how does an Rfam curator decide where the threshold is going to be, where they're going to set the threshold, the score threshold, above which they're saying, Rfam is saying everything above this score is a real match, and things below this score are perhaps not. We're starting to find false positives. But I guess that's part of a larger scheme. So what Rfam is doing is it's starting with an alignment, a multiple sequence alignment of related sequences, related non-coding RNA sequences, annotated with a structure, with a secondary structure, as I said earlier. They are then searching, Rfam then searches that, well, builds a covariance model from that alignment, which represents both the sequence conservation and the structure conservation, and then searches its collection of genomes. And then the step that Nancy's just described comes in, where they're choosing a threshold, say, of all of these hits, very long list of hits that we've got to this model with all of these scores, this is the threshold above which we think things are real and below which we think things are not real. There might be another step then that comes from that, which is a sort of iterative step where because you might have pulled new members into the family. So you might have discovered new members which were not in the alignment you started with. And you can align those sequences against the model and then go around again. So you now have a new starting alignment annotated with a structure, which will hopefully be better, and we'll find even more members. And so it can be a sort of iterative process. And the bit that Nancy just described in detail there was how do you choose a score? But that's part of a larger scheme, I guess.
Yeah, I can go a little bit. So I think there is not a region that is best represented for non-coding RNA families. Maybe you are talking about riboswitches because this happens in bacteria, and bacteria have a lot of regulations in the cis. And so that means that 5' regions are going to be easy to find non-coding RNA regions, but not necessarily in eukaryote. So I think, or not necessarily in viruses. So I think we really don't make that analysis yet for the Rfam families to really know which of the regions is more represented than the others in the families.
I will say one. Oh, sorry. Go ahead, Sam.
Sorry. I was going to say we find non-coding RNA, structured non-coding RNAs everywhere in the genome. I guess the vast majority of them are between otherwise annotated genes. So they're between protein coding genes, but they do also overlap with protein coding genes. So as Nancy just said, as the question asked and Nancy just said, there are some structured RNA regions in untranslated regions of protein coding transcripts. There are also some structured RNAs in introns of protein coding transcripts. So microRNAs and snRNAs are good examples of that. But actually the vast majority of different types they're genes in their own right, and they are found between other genes, so between protein coding genes, and therefore can be everywhere in the genome, scattered throughout the genome.
And I was just going to say that since we use the UniProt reference proteome set, these are generally well annotated with proteins, and so you can see, the protein coding genes and the non-coding RNA genes on one genome, and don't have to try to juggle coordinates between different versions and these sorts of difficult and irritating tasks. Yes, so thank you everyone for joining. Yes, it's been wonderful. Here is some information on some upcoming webinars. So the next one is on the 23rd about some biostatistics, and then on the 24th about human missense variation in ProTvar, which I think is a new resource the EBI just has out, which is pretty neat. Although this webinar is recorded and will be available online at that link, as well as all the other EBI sites. Finally, there will be a survey that I think launches very shortly. Please fill it out and let us know, and so we can try to improve the webinar for next time that we host this. Thank you very much, everyone.